Hi, my name is Nimrat Chatterjee and I'm an assistant professor here at the Department of Microbiology and Molecular Genetics. I'm an associate member at the University of Vermont Cancer Center and a member of the Vermont Center for Immunology and Infectious Diseases. Today, I'm very excited to show you what we pursue in my lab. We are very interested in understanding how the genomes or DNA of cancer cells differ from the normal cells. In other words, why do cancer cells continue to divide and proliferate while the normal cells don't? My research passions ignited when I was pursuing my PhD in the Wilson Lab at Baylor College of Medicine, where we investigated how environmental stresses impact repetitive DNA. And then later as a postdoc at MIT in the Walker Lab, where my research vision materialized, where I wanted to focus on how cancer genomes differ from normal cell genomes. So the two key questions that we're interested in answering are: how is it that the cancer cell genomes are maintained differently than normal cells? And more important, why do cancers keep coming back? Why is it that a previous successful treatment regimen no longer works? Normally, when a DNA is damaged, a specific DNA repair pathway comes in to fix it. But sometimes the damage cannot be repaired. And in those instances, a specialized group of proteins called translesion synthesis polymerases comes in to tolerate the damage. So what is translesion synthesis? In translesion synthesis, a group of proteins come together to bypass a DNA damage, whereby they try and insert anything across the damaged DNA and also help extend past that damage. And in doing so, if there are incorrectly inserted nucleotides, such as T over C, these incorrect insertions then become mutations in the next round of replication. Interestingly, cancer cells can use the same proteins to mutate and resist cancer therapy. To address some of these questions, we use basic tissue culturing techniques to study the biology of cancer cells. We also employ some of the tissue culturing techniques to study which combination of drugs can kill more cells. At the same time, we use several techniques to visualize cells under the microscope. In the same parameter, we're also very interested to examine how gene expressions changes in response to new combination of drugs. Together, these techniques give us a vision of how to target cancer cells to increase more killing. My lab fosters a very collaborative, focused, and a happy atmosphere to do science. I welcome you to contact me with more questions. We at the University of Vermont Cancer Center promote diversity, research excellence, and patient care. Thank you.